Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Most people think that you can replace sunlight with a vitamin D pill or supplement, but biologically, that is not even close to true. Full spectrum sunlight does far more than just increase vitamin D levels. It affects multiple pathways involving metabolism, circadian rhythm, and even cancer biology. In this video, we'll break down how UVB exposure leads to vitamin D production, how vitamin D and its metabolites function in the body, and why sunlight may have effects that go far beyond what a supplement can provide. So without further ado, let's get into it. So this is going to be a multiple part mini series about vitamin D and why it's so important, but also why it's so important to get it from the sun instead of getting it from a pill. And so this first part is going to be about why you should get it from the sun and not a pill. So before I teach you what I want to teach you regarding vitamin D synthesis inside of our skin, I want to go over quickly the solar spectrum and the electromagnetic spectrum for light specifically. So on the far right here, we have an invisible type of light. We can't physically see it with our eyes, but it's there. And that is ultraviolet light. And it's broken down generally into three categories: ultraviolet C, B, and A. And it's believed that we're not ever exposed to UVC because of the atmosphere and it blocking it. So therefore, we are only really exposed to, on a regular basis from outside in, UVA and UVB. And both of those spectrums can perform different functions for the body. And then there's what we all are familiar with, the visible spectrum: violet, blue, green, yellow, orange, red, etc., right? And then there's another on the far left side of this table. There's another part of the spectrum, which is also invisible, called infrared. And infrared is also broken down into several different things. So near, mid, long, or far infrared. If we were to take this graphic and superimpose it onto the solar spectrum, and this is very likely a solar spectrum that is going to be part of the year where ultraviolet is present, because the ultraviolet part of the spectrum is on this graphic here. But to make the point is that the sun is going to produce anywhere from about 270 nanometers, and what it reaches the earth is what I mean. And it's going to go all the way out to, I've seen graphics go out as far as 10,000 nanometers in far, far infrared. And what I think was interesting about this table is that it says here 52% of all of the solar spectrum is near infrared. And that is what's here, about 700 all the way out to about nearly 3,000 nanometers. So this is near infrared. And so huge percent of the solar spectrum is that near infrared. And how does this differ to other sources of light that we may be exposed to? Well, if we were to take a red light panel, for example, or a photobiomodulation panel that's LED in nature, and let's say that panel has 630 or 635 and 670, it's going to be a spike like this. Let's say the target wavelength is 635 in the red or 670 in the red. You're going to see a spike where you're going to have most of the energy is going to be at 630 or 635 and 670 respectively. But because of the nature of an LED, it's going to be off by about 10 nanometers on either side of that spectrum. Now, this is what is being used for therapy because we know certain wavelengths that have been studied have more benefit than others. And that's not the point of this video. The point of this video is that it looks nothing like what we're exposed to when we're outside. Is 630 on here? Yes. Is 700 on here or 670 on here? Yes. But it's part of a larger holistic way of getting light. And so when we are outside and we're at the time of the day and the time of the year that ultraviolet B is in the environment, then that ultraviolet B will take a precursor of vitamin D called 7-dehydrocholesterol, and through a non-protein or non-enzymatic reaction using ultraviolet light, it will convert that cholesterol derivative to vitamin D3. And the way that a lot of us learned about vitamin D metabolism in undergrad or in medical school or whatnot is that it has a very certain pathway that it goes along. This 7-dehydrocholesterol gets converted to vitamin D3 or cholecalciferol, and then that cholecalciferol goes to the liver and through an enzymatic reaction gets converted to 25-hydroxy vitamin D. This is the level that we check in the bloodstream for the vast majority of people looking for your adequate or inadequate vitamin D status, 25-hydroxy vitamin D. And then 25 hydroxy vitamin D is converted at the level of the kidney to activated vitamin D, or calcitriol, or 125 hydroxy vitamin D. We don't generally check this lab because we assume effectively that if you have normal kidneys or relatively normal kidneys, this conversion happens if you have adequate amount of 25 hydroxy vitamin D. So therefore, we would only check this on people who have chronic kidney disease or even like end-stage renal disease, like you're on dialysis. And in those patients, even if you have good levels of this pre-vitamin D, because you don't have the enzyme necessary in the kidneys because they're damaged, to make this, we actually supplement this as a drug to patients called calcitriol. And then this is thought to be the activated vitamin D, and it's going to have target tissues that it has effects on. And it does. I'm not trying to say that this 125 hydroxy vitamin D is completely useless, but it's not the entire story. So again, here we have sunlight, ultraviolet B, right? It's going to take that 7-dehydrocholesterol. It's going to convert it to cholecalciferol or vitamin D3. That's going to go to the liver. The liver is going to convert that to 25 hydroxy vitamin D. And then that 25 hydroxy vitamin D is going to go to the kidneys, and the kidneys, if they're functional, will make biologically active vitamin D, 125 hydroxy vitamin D. But there's a big problem with this dogma, as you may say, or understanding that's taught all over the world, is that that's not the entire story. So there's a paper published here in November of 2019, and it's titled Vitamin D Metabolism Revised, Fall of Dogmas. And it says here that the dual origin of vitamin D, discovered about a century ago, first evolved into a rather simple metabolic schema constitutive of 25 hydroxylation of vitamin D in the liver to produce 25 hydroxy vitamin D, followed by a tightly regulated 1 hydroxylation by a unique CYP27B1 in a unique organ, kidney, to regenerate 125 hydroxy vitamin D, or calcitriol, or activated quote unquote vitamin D, as a ligand of a nuclear receptor, vitamin D receptor. All these metabolites are transported by a single serum binding protein and are finally catabolized by a unique, nearly ubiquitous CYP24A1. The present picture is much more complex, with a large number of enzymes expressed in a variety of cells. Most of these genes contain genetic polymorphisms which may alter their function and are regulated by hormones and or metabolic signaling that can vary in different tissues of the body. Finally, the vitamin D endocrine system regulate a large number of vertebrate genes. These recent findings reveal that vitamin D endocrine system is much more complex than initially thought and remains incompletely understood. I clipped out some snippets from this paper, and the first thing was that vitamin D is one of the most frequently used medicinal products in the world. Many people are on vitamin D supplements. Dietary intake is irregular, as few items contain vitamin D, and is usually well below human requirements. So its synthesis in the skin is the most important source of vitamin D. Although there are probably around 50 known metabolites of vitamin D, measurements of 25-hydroxy vitamin D or 25-OHD is clinically used to determine the vitamin D status. That's still used today. We still use 25-hydroxy vitamin D today. Whereas the serum 125D is used to access the biological activity of vitamin D endocrine system. We don't check that often clinically. Vitamin D esterification later says that based on clinical supplementation trials, only 1 out of 3 to 10 molecules of vitamin D, cholecalciferol, D3, is converted to 25-hydroxy vitamin D. So only 1 to 3 of the vitamin D molecules are converted to this 25-hydroxy, which we use as the gold standard to know whether or not you have enough vitamin D or not. The same is true for the conversion of 25-hydroxy into 125-hydroxy. The other 25-hydroxy molecule can be converted by these other CYP proteins to a large number of other metabolites. And that leads us to a paper that was published a year later, and it was titled Photoprotective Properties of Vitamin D and Lumisterol Hydroxy Derivatives. And it says here that these CYP11A1 derived compounds are produced in vivo and are biologically active, displaying anti-proliferative, anti-inflammatory, anti-cancer, pro-differentiation properties, since the protective role of the classic form of vitamin D, 125-hydroxy vitamin D, against UVB-induced damage is recognized. We recently tested whether novel CYP-derived D3 and L3 hydroxy derivatives protect against UVB-induced damage in epidermal human keratinocytes and melanocytes. We found that along with the activated vitamin D, quote-unquote, 125-hydroxy vitamin D, these D3 hydroxy derivatives and L3 and its hydroxy derivatives exert photoprotective effects. These included the induction of intracellular free radical scavenging and attenuation and repair of DNA damage. the protection of human keratinocytes against DNA damage included the activation of NRF2, regulated antioxidant response, p53 phosphorylation and its translocation to the nucleus, and DNA repair induction. And so, as alluded to in the prior article, there are about 50 roughly known vitamin D metabolites other than the classical pathways of 25-hydroxy and 1,25-hydroxy vitamin D, the quote-unquote active vitamin Ds, right? We now know there are more than 50 of these chemicals, and they are made in different parts of the body, and they are made under different circumstances and in different amounts depending on how active the enzymes that make them are. And what we know is, based off the abstract and also subsequent graphics, is that these D3 and L3 derivatives are also very biologically active. It's not just activated vitamin D anymore. That's the death of the dogma. So these D3 and L3 derivatives, which we don't measure, and we can't measure as of right now clinically, are operational at helping protect against preventing cancer and maintaining calcium and vitamin D, other vitamin D metabolite levels, and having antioxidant effects, and having immune boosting and anti-inflammatory effects, as well as DNA repair and anti-cancer effects from the DNA perspective. So my point is that this is one of the big reasons why you cannot replace what you get when you're exposed to ultraviolet B light in a pill, because you're missing out on all of these 50 other chemicals that have anti-cancer effects and other disease-modifying effects that you would not be getting in a pill. If that wasn't enough, just from a vitamin D perspective, we also know that depending on the time of the day, different color temperatures help signal to our body what time of day it is. Classically, this is thought to be done through the melatonin system. And that is true. It is done through the melatonin system, but it's not only done through the melatonin system. It is now known that vitamin D helps to correct and maintain our circadian rhythms. And why would we care about that? Because the circadian rhythm is directly related to how our body responds to the hypoxia genes, hypoxia-inducible factors. So we now know that these hypoxia-inducible factors are responsible for metabolic reprogramming and pretty much all of the development and maintenance and progression of cancer: angiogenesis, invasion, metastasis, proliferation, inflammation, the blocking of apoptosis or programmed cell death. And what do we see that vitamin D unilaterally can do? It can block all of those things, right? And this is a graphic looking at calcitriol specifically, or 1,25-hydroxy vitamin D. But we now know, because of further research, that when we're exposed to the sun and we make all these other 50 vitamin D-like chemicals, these D3 and L3 hydroxy derivatives, they also have similar and diverse anti-cancer effects in addition and above and beyond what the quote-unquote activated vitamin D can provide us. So these different articles and different mechanisms and different chemicals that are made are meant to be firepower for me to help persuade you that what you're taking in your pill that makes your 25-hydroxy vitamin D level on your lab test look pretty is not the whole story. And you're missing out on a lot, which is why I am very much so pro full spectrum sun as the first line treatment for vitamin D deficiency, as well as other deficiencies that we will see that the sun also corrects. If you like videos like this, please like, share, and subscribe. Until next time.